Good morning to everybody. Good morning to the large family of MD Anderson from here, Houston, from United States and all over the world. Thank you for being with us today at 11 o'clock because sometimes in life and science it's good to make changes. From next Friday, again, we come at our usual hour of 10 a.m. Enjoy Science, it's a webinar for all of you, for the ones who love medicine, who love science, and for the one who love MD Anderson. And it's organized by Dr. Giulio Draeta, and I'm co-hosting it with my friend and colleague Alessandra Ferraioli. Of course, I thank you as many times as I can because without the attendees, webinars make no sense. And I thank also to Liz Kent, Ellison Gart, and Valerie Jimenez because without their hard work day by day, this would not be possible. But the most thanks go to the speakers. All of you who support Enjoy Science have done a wonderful job, and everybody is happy about the talks. There are more than 7,000 attendees till now, and today we have a big pleasure. We have a big pleasure to have as a speaker a person who is a true pioneer in science. He is a visionary in science, and I can say now his name, Dr. John Weinstein. John, it's really a person that all of us, we know, we meet and we collaborate in MD Anderson. Probably most of you know that he is a PhD and an MD. He finished Harvard, Stanford, but not for this he is famous. He is famous because really he had a wonderful vision in science when nobody was thinking in this way. Early 90s, John Weinstein is a pioneer who starts a multi-omics approach in a famous panel of cell lines named NCI60. He was working in NCI at that moment, in which he has done genomics on RNA, DNA, and proteins. Now, of course, all of us are doing this, thinking how simple it is, but yes, it was a person who teach us how to do it, and this is John Weinstein. Not only, but the clustered maps, heat maps that we are doing, red, green, and all these colors that no, there are day-by-day science now. Everybody loves them and knows them. They were started also by John Weinstein in the same period. And he continued doing wonderful job in bioinformatics, and he became 2008 the chair of bioinformatics department in MD Anderson, and I praise him very much for the fact that he was able to put the seed of developing wonderful young scientists, bioinformatician in MD Anderson. So we can say now that in our place, it's also a John Weinstein School of Bioinformatics. So with this said, let's pass the show to John. John, thank you for being here. It's a great pleasure, and we are looking very much to your very interesting talk. Good luck.
Thank you very much, George. I am very glad that this is being recorded because then I can play it for my wife and maybe I won't have to take out the garbage or maybe I'll have it for my tombstone. Thank you very much. Before I start, I just want to remind everybody, especially those in the Houston area, of this upsurge in COVID-19 disease, hospitalizations. It's not just a matter of testing, as indicated by hospitalizations. For example, as you think about how you live your daily life, both in work and at home, pay attention to what was said by a Texas politician, Marco Rubio, a couple of days ago. He said, wear a damn mask. So wear a mask and please take care of yourselves. You're very precious for the work you're doing in cancer, as well as to yourself and your family. George, you must be psychic to have invited me for this date. And I must have been psychic in deciding to talk on the subject I'm going to talk about. Both of those hold because of what I am going to show you in the next slide, which was that 2 days ago at its virtual annual meeting, the American Association of Cancer Research conferred the Team Science Award for 2020 on the Cancer Genome Atlas program. This was a seminal event which in a sense capped 12 years of maybe 14 years if one starts from the beginning of the process of effort by so many people in that program. And here are some of the people who are honored. I would like to say just a word about Anna Barker. Ann Barker, who was, in a sense, my guru in this subject, and who has supported our work and has shown an imagination and creativity and ability to think big, which has made the project successful to a considerable degree. And also thinking big, Eric Lander, of course, a collaborator along the way with myself and my laboratory. Also would like to thank Margaret Foti, CEO and spearhead of the AACR, as well as the directors of TCGA in its mature phases. Kenneth Shaw, who you probably know if you're here at MD Anderson, is in genomic medicine and associated with IPCT, and also JC Zenklusen. Both of them have the characteristic that they also think big, they think concisely, and they know how to kick ass when it's necessary to herd cats, as is certainly necessary with large egos in a project like TCGA. More locally, if I can be parochial about it for a moment, three of us in our department, bioinformatics and computational biology, were awardees of the Team Science Prize as part of TCGA. And though they were the only three, for whatever reason, who were specifically mentioned, I'll also point out that others in our department have been deeply involved in TCGA and the projects related to it that I will be discussing with you. Also, our software engineering team, our bioinformaticians, bioanalysts, trainees, it's been a team effort. Other members of the MD Anderson contingent who've also been honored in that way are shown here at the bottom of the slide. And I'll point out that 4 of them have been members of the Genome Data Analysis Center that the department established that I'll mention later on. Now, that having been said, the way we used to do science, I would say, was cottage industry. Before, let's say, 1985, 95, and the advent of microarrays, it was more or less one gene, one protein at a time for half a career. And incidentally, you'll probably see that this is a deep fake image. It's not Photoshop, but well, it's still representative because I once upon a time was a lab rat. At this point, the technologies have advanced so far that though I claim it's because I don't have time, it's really true that no one would allow me in the laboratory anymore. But nonetheless, this is the way we used to do it. But now, of course, we have high throughput technologies, single cell sequencing, instrumentation of all kinds, very impressive, and things that we are doing, including the 5 of us in our department who have wet labs in addition to dry labs. Well, this is on the informatics side, paper medical records, and now EPIC is representing our electronic medical records, and under a new CTDO, David Jaffrey and Andy Futrell going steps beyond even this sort of medical record to establish the infrastructure for sharing of data around the institution at all levels in research and clinical data. Now, this is a slide that's going to be recurring here. This is really the thematic slide of my talk. It's an attempt to look at the phylogeny of projects in molecular profiling of cancer and painting the molecular picture. Now, it's not complete. It's a work in progress. You'll see some dates. You'll see question marks, which I will fill in for you over the next 30 or 40 minutes. So let's start with 1998 to 2001 Human Genome Project. I put it in that time frame because essentially all of the sequencing was actually done in the final stages. And that's for a reason. Here in 2000, we have what's known in many quarters as the Treaty of Bethesda. The two sometimes warring factions, Craig Venter and the Collins contingent at the NIH represented here, parallel papers in Science and Nature. This was a time when even presidents were interested in and excited by science. This was a great occasion, even though it represented just a draft of the genome. As you know, it was in quotation marks completed in 2003, but bedeviling those of us who are trying to keep up in terms of our scripts in bioinformatics, the builds keep coming on over and over again. So, Human Genome Project, but I'd like to go back even further to what one might call the pre-Human Genome Project. In 1986, Charles DeLisi, who had been my laboratory chief at the NCI and had since gone to the Department of Energy, had the crazy idea of sequencing the genome of humans. The reason that was crazy is no one at that point had the slightest idea how one could ever do such a thing. The technologies simply didn't exist. It was also viewed in many quarters as crazy because of the notion that it would take away from the R01 pool for individual science. It would never work or never be as important as people like Charles and Renato Del Becco were saying that it would be. But of course, we know now that it's at the heart of everything we do. It's the basis for so much of our science, no matter how we're approaching the science in laboratory or on the computer. Let me switch now even earlier to the NCI 60 drug screen that George mentioned. This was established principally by Robert Shoemaker and Kenneth Paul. I had the privilege of working with both of them. Kenneth Paul unfortunately died much too early. We treasure his memory. Well, the story of my involvement is one of those bifurcations in life. As you know, there are almost chance incidents that can change your trajectory, just as there are chance incidents that could change, for example, the stability of the solar system, or could determine whether a particular evolutionary track is faced. I'm fascinated by the notion of bifurcations and complexities, complexity theory, but that's for another lecture. So the NCI 60 drug screen, 60 cell lines, the NCI 60 used to screen for anti-cancer agents. There was a June 3rd of 2001. I was staying up late. I wasn't going to go to grand rounds the next morning, but Bruce Chabner, director of the clinical division, was giving grand rounds. So I pulled myself out of bed, and I did, and the rest changed my career. Up to that time, I'd had a perfectly good career. I had just organized a Gordon conference, bunch of science papers recently, but I felt like a graduate student preparing to do something, but I didn't know what it was. Well, it turned out to be omics. And I say this because I know many of you are young scientists listening and trying to figure out what it is that's your passion in the long run. Sometimes it comes early to you, sometimes it comes late, but whenever it does, follow it for sure. One of the things that came up in Bruce Chabner's lecture was the NCI 60. I was working at that time on the side on using neural networks for protein structure prediction. Good results. And it occurred to me in his lecture that we might be able to use what at that time passed for artificial intelligence predicting mechanisms of drug action in the screen. So I went up to Bruce and he introduced me to Ken Paul, who worked with me. And by that night, we had the system set up. for neural computing in cancer drug development. Here is a truly primitive neural network. Well, it didn't seem primitive at the time, even though it had only one layer, nothing like the convolutional networks such that are tools of the trade now. But nonetheless, it was exciting to me and led me to get more interested in databases that were coming from the NCI 60. So here's what it looked like. At the top is the flow in a portion of the NCI drug discovery program from the inventory, 60 cell lines, a very large effort for the time really to do the testing to produce the database A for activity patterns across the 60 cell lines. Note that those then can be interfaced with the database of molecular structures. We developed sets of molecular structural features to do that. And then on the other side, having to do with molecular profiling, the characteristics of the cell lines. And the possibility also of adding more samples if one wanted then to extrapolate from the ones that existed. So to make it explicit, for correlation of structural features with activity, we put together information from these two databases. For correlation of activity with molecular structures, these two. And as a matter of fact, we were able to look across from structures through activities to cell lines to determinants in those cell lines. So that then can be viewed, if you're more mathematical in topological terms, looks like this in terms of the matrix and matrix algebra. So that was how we got started with the NCI 60 profiling. And I'm going to spend some time on that for a particular reason, that it was in a sense a 60 square chessboard, if you'd like to use that terminology, or a playground, or a model system with which to work out many of the rules of the road for these technologies for profiling and for the consequent multivariate informatics. What made the profiling possible was the super tech in my that I had at that time, Bill Reinhold, who's since gone on to play a considerable role in the NCI's intramural program in this regard, I'm very happy to say. Bill was one of those people who was creative, productive, and also, unlike me in the laboratory and unlike the collaborators we tended to have, never made a mistake, never mislabeled anything. And so he was just the right person for the job. We worked out the ways of developing DNA from the 60 cell lines, RNA, protein, microRNA, and that was put into a number of what I called integromic, and these days more familiar as multi-omic profiling. At the top, you see the DNA measurements we did over a number of years, then the RNA. Long non-coding RNAs weren't in focus at that time, I'll point out. Proteins, small molecules, and then validation studies by what we had at the time, RT-PCR, siRNA, no CRISPR, of course, and tissue arrays. So let me just say something about the 2D gels, because there's a lesson in it. This was a study in which we did with Lee Anderson, then at Large Scale Biology, duplicate 2D gels across the 60 cell lines, developed a version of MALDI-TOF for spot identification, looked like this in the master gels, indexed 1,014 spots overall, wrote the paper, gave it to an editor at a journal that shall remain nameless. It was returned to me without review. The only comment was, where's the hypothesis? This brought front and center what's a continuing theme in this field, which is that on the one hand, we're used to the idea of detective work, one gene, one protein at a time, very incisive work, following up particular hypotheses. In my laboratory, we certainly have done that sort of thing, but we also want to come from the global end using omics, and I'll come back to that a bit later. It's my feeling that we need a combination of hypothesis-driven and omic research to get where we're going. Even though by the time, this time, of course, we know the importance of the Genome Project as an essentially unbiased look over large territory. It is sometimes forgotten by reviewers and by editors and by site visitors and by study sections how important the unbiased large data sets can be. And certainly in the 1990s when we were doing this work, it wasn't recognized in full form yet because the Genome Project hadn't yet eventuated. Well, we did our thing with the heat maps, and where's the hypothesis was still the result. Let me just run through, I'm just really going to just flip through these slides just to show you the breadth of it, some of the techniques that we used and kinds of data that we accumulated on the MCI60 for this multi-omic, integromic sort of analysis. First of all, what now, at least at MD Anderson, is called reverse phase protein arrays. Worked with Lance Leota, Chip Petricoin on that technology to develop it and applied it to the NCI60. Array CGH for copy number changes. Also worked with Joe Gray in the early days on that. Nimblegen arrays for the same thing. This was bisulfite sequencing results for methylation of the DNA and the consequent informatics, which were developed by So-Young Kim, shown down at the lower right. Spectral karyotyping, which defined the importance under Ilan Kirsch and Arushki of hypervariability in the genome. Phil Lorenzi, who, if you're at MD Anderson, you probably know as director of the Metabolomics Core, which is a function of our department. Phil was a postdoc with me at the NCI. We got very worried in terms of the quality control of the NCI60. He did the STR fingerprinting and confirmed what we could see even by looking at cells in the dish, that there was some confusion that had to be corrected. Speaking of Phil and metabolomics, this is the opening of the Metabolomics Core, and I mention this because quite a number of you will be using it if you aren't already. Peter Pister, president of MD Anderson, shown there talking with Phil in the upper left. And if you look at the lower left, that's me pretending to get some actually productive work done. I brought with me from NIH a custom of funny hats, which is, so this is not normal gear for our laboratory. So this is the kind of thing that we were doing. I take a comprehensive molecular profile of the NCI60. As I say, thinking of it in part as a model system in addition to what might come out of it for drug discovery. And everybody, I think, has a favorite paper that they've written, one that they're proud of. This is the one that I'm most proud of, from 1997. I call it Information Intensive Approach to the Molecular Pharmacology of Cancer. We didn't yet have in it the consequences of the revolution of microarrays, let alone molecular sequencing, but we had cobbled together what we could get from PCR, et cetera, and mapped out those 3 databases that I showed you in a previous slide. This leads me to the question of the omic world. At the time we were doing a lot of this work, genomics was a word and proteomics is a word. I was amused by the fact that we have genomics and then genetics, if you're thinking one gene at a time, proteomics, and we don't have protetics, but that's just a nuance. Now this slide is one which would be better delivered in person when people could laugh and be heard. Well, some of the comics in late night are getting used to the fact that they can't get the feedback of laughter. Others, like Bill Maher, for example, have introduced laugh tracks in order to do their stand-up. In any case, I will assume for purposes of my morale that at some point and during this slide you'll chuckle just a bit. So in addition to those two, I thought we should have phenomics and kinomics and chomics and metabolomics and immunomics and toxicomics and clinomics. And then the compound forms like functional genomics and structural genomics and pharmacogenomics and pharmacometabolomics. And I was lecturing to a group at NIEHS on ecology who were doing some profiling, or actually thinking of doing it. And we had ecogenomics, ecoproteomics. I figured that there should be a single term that encompassed both. So I suggested economics, but they were afraid of being confused with a dismal discipline, so they didn't like that much. So we tried ecomics, a little better. Sometimes, however, it looked like ecomics, which was not so popular. Well, all of this is fun and games, but in fact, ohm is from the Greek for an abstract entity, group, or mass. It's sanctified by Webster's unabridged dictionary as etymological purity, therefore, and I put a piece in Science to make that clear. Well, again, this is the flow in the NCI, in the drug discovery program. And the first thing is to capture all of that information. This is a screenshot of CellMiner, which is a program we started as a relational database in order to make the data sets available to the broad community. That's been carried forward since I left the NCI by my former laboratory under the aegis of Bill Reinhold and also Yves Pommier, the chair of the department. This is a resource that you can query for the NCI 60. And what that brings up is the informatics. So let me switch to that from the profiling per se for a couple of minutes. This was the kind of thing we were working with, let's say a gene in a space of 60 cell lines, 60 dimensional. What do we do? One of the things we have to do is to reduce the dimensionality to do anything sensible with the data. Variety of techniques from classical statistics for performing various functions. This is a set that we used at one time or another, the ones in black at the time of the NCI 60 work, and the others later on, PCA+, RBN, EB+, which I won't take the time to explain, were developed by Rehan Admani, and next generation clustered heat maps already been discussed as static forms, but now we've developed them as highly interactive exploratory environments, and I'll say a little bit about that later on. In addition to those techniques, the computer-intensive statistical methods, including the more recently developed TISNE, UMAP, and such, some of them based on AI methodology. And then finally, speaking of that, AI machine learning, the very simple neural nets that I'd started with, but now, of course, to the point of the convolutional neural nets that we're using from TensorFlow or very interestingly, huge numbers of scripts that have appeared in bioconductor and CRAN libraries and other areas. Another lecture that I really enjoy is talking about those sorts of informatics, in particular adversarial learning networks and generative adversarial networks, maybe another time. So the only thing is I'll talk about now the clustered heat maps. This is something I didn't devise. It was a reenactment by a photographer from MD Anderson's Promise magazine. Sometime in 1992, we had, and this is with then postdoc with me, Tim Myers, developed a way of expressing all these data on drug activity, molecular targets, drug structural features. And for even 1,000 drugs, that came out to be 28 feet of data, as shown here. So by stages, we then ended up reducing it to the color-coded clustered heat maps that we are so familiar with, in this case for drug candidates against potential biomarkers, not yet microarrays at the time of this figure being made, but subsequently much larger matrices, now up to 100,000 on an axis, for example, for single cell studies. The first success, medically speaking, of clustered heat maps was in 1993. It was taken by collaborator Tito Fojo to a go, no-go decision conference in Switzerland for clinical development of oxaliplatin, which you probably know is still a standard agent for treatment of colorectal cancer. In this figure, you see on the two axes, it's symmetrical, platinum compounds clustered by activity, platinum compounds that had been through the screen. At that time, what was most in focus, of course, was the cis-carbo family. What you see here is along the axis a very well-developed set of families of higher potency, very much associated with particular structural features of the platinum compounds. The diaminocyclohexyls, including oxaliplatin, also turned out to be selectively more active in colon cancer, and that then tipped the balance for development of that agent. Well, Cancer Cell Line Encyclopedia and its British associate GDSC clearly followed very, very closely the pattern of the NCI 60, but with much larger numbers of cell lines. This is way out of date. This is from actually 2012 from a News and Views piece then, as opposed to the NCI 60, which has only 59 or 60 cell lines, depending on the vintage, but more than 100,000 compounds tested plus natural products. And also then, of course, we can be thinking towards clinical tumor sets. So I view the CCLE, GDSC, and NCI 60 as each having some proper functions. Well, the human brings us to TCGA, 33 tumor types, and the Pan-Cancer Atlas Project taking the data across and analyzing across those 33 types. I was invited to speak to the executive, the NIH, the NHGRI, NCI executive committee that was planning TCGA, a number of other interactions, in order to try to indicate what lessons we'd learned from the task of profiling the NCI 60. Well, of course, it's much more complex to think about doing that in humans as it is anytime the clinic is involved. A heroic effort for sure. And again, as with the Genome Project, there was a lot of discussion as to whether it was going to be a waste of resources that should go into the R01 pool or whatever. And it took a massive effort in order to make it work. But it did. It produced databases that are being mined, I assume, by many of you who are on this conference call. It called for molecular profiling of about 500, but it turned out to be more in some cases and fewer in the rare tumors of each of 33 different clinical cancer types, and a large variety of measurements were made. It started off to be just DNA sequencing, but then pretty soon it became clear that some phenotyping at the DNA and RNA levels should be done. And then the proteins came in and the epigenomics. There was a famous conference in which those who were doing the epigenomics pleaded successfully for that to be included, which was done very successfully. So let me just tick off the kinds of measurements that came into play. And then more recently, techniques like ATAC-seq and others of more recent vintage. Well, we were fortunate enough to get funding to establish a genome data analysis center for MD Anderson, centered in our department. We continued with that to present day under a rubric of either TCGA or of its descendants in the NCI. And this is the way I thought about the NCI 60, and then the way we came to think about the pan-cancer TCGA project, what I call the pan-cancer cube. One axis for tumor types, a second for genes or loci, and then a third for data types. I think it's pretty obvious now that this is a primary way to get to the point of doing multi-omic analyses, not just in terms of molecular profiling, but when one adds imaging, adds the demographics, clinical data, and the like. So how do we want to approach that in terms of software? Well, this is a smattering of the projects that we brought to the point at which they're highly engineered for use by biologists and clinical researchers, as well as by informatics specialists. These are all on our departmental website. I'm not going to talk about any of them except the next generation clustered heat maps, just for lack of time, though I also will actually point out the standardized harmonized TCGA data, now expanded beyond TCGA, is the best place, I think, to get analysis ready, batch corrected, refined data at the level 3, that is at the gene level, not at the sequence level. And then also I'll mention that batch effects analysis was something that we did in near real time, saved some embarrassment when glitches had arisen in the data set as each of the 33 projects came out. One of the things we did was to establish next generation clustered heat maps in a portal of number of 100 for TCGA and TCGA pan-cancer. When I say next generation heat maps, I mean that you can zoom, navigate on them like on a Google map, link out to a couple of dozen different external resources, a statistical toolbox, etc., that I'm showing you here. Thinking of these interactive heat maps, which have been implemented on the cloud, in servers, in containerized form, in Galaxy, for those who are into such things, surrounded it with a set of toolboxes for annotation, chromosomal characteristics, statistics, clinical, biological, as an exploratory environment in the way we thought of it. And then we've done some other things like this for single cell next generation clustered heat maps. We included, in addition to the heat map, cell-wise information in the form of UMAP or TISNE, or you name it, kind of scattergram that's used particularly in the single cell field, but also more widely. If you think about it, those representations hide the gene by gene information. And on the other hand, pathways hide the cell by cell information. All of the information in those senses is included in the heat map. So we've developed the machinery for moving back and forth fluently among these and other representations that are suggested here. The team that's been working on this, you'll note below, including those in the software engineering enterprise. We've also then taken this, and this is typical of what one would like to do with many of these software engineering tools and resources, is to create interfaces of them with the environments and tools that others have produced. This figure shows both existing and proposed versions with the ITCR, the Information Technology for Cancer Research project, and also with others. And that brings me to the final set of boxes here. 1,000 flowers have bloomed. This is just a selection of the large atlas projects that have been undertaken by the Cancer Institute, by other institutes at the NIH, by the NIH Common Fund, and so forth. So here, and I don't expect you to read all of this, and I'm certainly not going to go through it, since these slides will be available to you. You may want to go back to this just to remind yourself of some of these entities. Target for childhood profiling, Alchemist, lung cancer motivated, for example, by the effects of the first generation EGFR agents, and then those for downstream recurrences, GTEx, normal tissue expression project, the human cancer, the human cell atlas, using single cell studies extensively. I put PCOG here, pan-cancer analysis of whole genomes, reminds me that in February of this year, a set of 13 papers came out together in Nature journals on COG. Very interesting from the point of view of what one can get out of whole genomes. And then an additional paper is past proof stages on the informatics tools from PCOG that will be appearing in Nature Communications. And the Human Tumor Atlas Network, again, extensive single-cell studies, as is the case with Precancer Atlas. And then internally at MD Anderson, DECODE, spelled with a 3 rather than an E because DECODE itself was already taken. This is for data-driven determinants of COVID-19 oncology discovery effort. Another of the tortured acronyms, but a huge and really very impressive program spearheaded by David Jaffray and Andy Futrell to corral information of many types, clinical and preclinical, molecular profiling included, at MD Anderson in the service of COVID-19 research. I'm not going to have time to go into any of these latter-day projects in detail. That would, again, be a separate lecture, and I think it's great fun and also exciting to see what's coming down the pike, but that would have to be left for another time if I were at some point invited back. Hopefully, COVID-19 won't be threatening us as it is now, long enough for that to be the case. In any case, this is the way the Human Tumor Atlas can be looked at. And then, of course, there are the incredible new technologies arising, it seems, daily. This is from Gary Nolan and his Fertile Mind and Laboratory, ways to make highly stained 50 to, he would say, 120 antibodies cross-linking to cells for histological dissection of what's going on across tumors. Very interesting work presented in the virtual meeting of the AACR last week. For those who are interested in the informatics, I'll just mention that I was asked by Bob Bast here at MD Anderson to write a chapter for one of the medical cancer medicine textbooks. I thought, since he gave me a year to do it, that would be easy, but I found that it hadn't been done before. There wasn't a good model for it. Nonetheless, here it is. If anybody would like a PDF of it, email me and I'll be happy to send it to you. Well, all of this reminds me that what we've been doing, in a sense, is drinking from the stream of molecular data. Back in the 1980s, one might say it looked something like this. By the mid-2000s, by 2016, and then my way of looking at it is it's just an unhandleable torrent of information that we're coping with in the informatics domain at this point. It's a labor of love and of tremendous excitement for so many people in the field. And from my perspective, there never has been as rich a time to be in the biological sciences and the biomedical sciences in particular than there is now. So a few generic conclusions from this talk that you might want to draw. First, the NCI-60 project was the first large-scale public multi-omic integromic molecular profiling effort in cancer. And as such, it provided this 60-square chessboard on which to work out many of the technologies, workflows, and informatics tools would later be applied in more mature form to TCGA and now to its descendants. And I say in much more mature form because all of these fields, both in the laboratory and on the computer, have changed so rapidly and with so much refinement that again, I can't help but use the word exciting. TCGA and its contemporaries have in turn provided a template for the new wave of more refined, sometimes single-cell studies on tissues from clinical trials. I emphasize on clinical trials because the new work of the NCI and the other institutes is almost entirely now on well-defined sets of cancers with more clinical information than was available to us in TCGA, and with fewer questions about possible bias in selection of tumors for study. Omic and hypothesis-driven research don't just complement each other, in my view, they synergize in our accelerating attempt to understand and manage cancer. We can't do it without both. And even in a time when we're rightly focusing on COVID-19, the pageant of progress against cancer continues. It can be pointed out that in the US, there are about 600,000 cancer deaths per year. And so that does give us reason to try to continue the work even in the face of the threats posed by COVID-19. And finally, team science works. Maybe it's surprising, maybe it's not surprising, but there has been a steady shift from thinking in terms of it as a competitive process to thinking of one as the benefiting to all from cooperation. Partly, I think that's because so many techniques have emerged. There are so many problems to be solved that it's not so much a matter of a field focusing on the one or two questions that it's addressing and everybody having to pile in competition to address those one or two basic questions. Well, finally, principal funding support is shown here. We're very grateful to the NIH support and also to that of the Michael and Susan Dell Foundation, Mary Kay Chapman Foundation, that have made our work in this field possible. And I've already talked about the major players and what I was showing you today, but I can't help just pointing out the large numbers of people that we've worked with at MD Anderson, shown on this slide, and then both in previous life and continuing collaborations at the NIH. Again, you can take a look at this in the slide sets that'll be available if you wish. But with that, thank you for your patience. And if there is time remaining, then glad to take questions. George.
Thank you very much, John. Thank you. This was a wonderful lesson for all of us how, in fact, team science work. And certainly, we prize you very much, not only for the science that you present us, but to be fair, also by the fact that you acknowledge all the team members and all the people who marked your career in science, because in fact, without mentors and without the people in our laboratories, all this makes no sense. And let's start with a question. It said the naive questions, but I think it's very, very important one, how to define the threshold of gene upregulated and downregulated.
That is not a naive question. That's a terrific question. And as a matter of fact, it relates to one of the more common mistakes that I see in the informatics, which is to pay attention to the p-value, and let's say making a binary comparison of treated and untreated, and not to pay enough attention to the actual magnitude of the difference seen. From a mathematical point of view, if you have a large enough sample size, then if there's any stray difference doing an experiment on Friday as opposed to Saturday or breadth of wind, then if you have a large enough data set, you will asymptotically get an apparently significant p-value. But it may be absolutely meaningless with respect to the data. Think of a box plot. You're very often seeing, I'm sure, box plots in which there's a comparison and a p-value of 0.003. But on the other hand, when you look at the boxes, they're pretty much the same place, and the medians aren't very different, and the distributions overlap so much that it really doesn't give you any ability to predict, let's say, which drug is better. So I think the lesson in its simplest form is pay attention to the absolute magnitude and p-value in that sort of situation. And when you ask more particularly, how much difference does there have to be in the absolute magnitude, that partly depends on what you want to use the information about a difference in groups for. If you are satisfied to get just a slight edge in terms of a drug effect, a slight statistical edge, then your criteria won't be as stringent as if you really need a very large positive predictive value or the like. So yeah, that's definitely not a naive question. It relates to one of the most common mistakes that one sees in analysis of data by those who aren't trained in the informatics. And let me just say that in organizing our department, we have those who are really trained in the informatics and those who are who are mostly applying the informatics. And my feeling is that in our problems, when you have a heavy-duty multivariate statistical problem, it really has to be approached by someone who knows the basis for the tests we use. And then the biological interpretation should be done principally by biologists, and we've tried to bridge the gap with what I call bioanalysts. But even the simplest of our t-tests and Wilcoxon tests and such have many hidden assumptions that can be violated.
Thank you. And next question. Do you think we have the right platforms to share the data across different fields, in the world, to cross the boundaries of the specific scientific question?
That's another very interesting question. I'd mentioned the ITCR, the Information Technologies for Cancer Research Consortium, which is mainly developing tools and resources with a major attention to that issue of cross-discipline, cross-field analysis and interpretation. The sharing of data is now, in a sense, a field in itself. We are constantly making progress in the ability to do it. For example, you've probably heard of containerization. Docker and Singularity are the most common programs for doing it. One of the issues in sharing has been that if I wanted to share with you my scripts for doing one of these multivariate analyses, and allow you to put it on your computer probably has a different vintage of our program, or it has different issues related to the operating system, such that it won't work directly on your computer. Well, now containerization to a considerable extent gets around that, as does implementation on the cloud. My suspicion, and I'm certainly not alone in this, I don't mean to say it's original, is that we are headed eventually for a situation in which we will plug in to the cloud the way we do to an electrical socket these days, and that the squeamishness about private about confidentiality on the cloud will give way to just thinking of it as a utility with a set of tools, many of them transparent enough so that they can be used by non-specialists. There's much more that can be said, but I will stop there.
Thank you, John. Another question. Great talk, John. Such an amazing perspective. Would love to hear your thoughts about all the other omics that are now using your heat maps. Also, what is the future of visualization of these multi-omics as we move forward?
Well, another good question. I indicated the beginnings of an answer when I said that we were working with a variety of consortia to implement them in the context of their environments, or to think of it in the opposite way. For example, and I didn't emphasize the individuals with gene pattern is well known to many as an environment that's now used UCSD and the Broad, Joe Meseroff and companions. We've now integrated it with that. We've integrated it with the metabolomics workbench, with CBio Portal, which I'm sure some of you are using. So there's an attempt to integrate it as a tool, in a sense, as a plug-in, or to indicate, or to incorporate others' tools into the NGCHM system as plugins, from Seurat for single cell, and so forth. So whereas we used to think, when I say used, I'm thinking back just a couple of years to when we developed software tools, we used to think of them as more or less standalone applications that one might download on the computer. But now we're thinking of developing modules that then can be spliced together as pop beads using the APIs that have been developed along the way to mix and match across applications and across disciplines. When the question asks, what about the heat maps used in other disciplines? Yes, for example, in terms of imaging, incorporated them into the imaging databases with, in particular, the IVGAP brain imaging databases, and we've incorporated them into the metabolomics. We're now in a collaboration with Jennifer Wargo and the microbiome community, both from the metabolomics perspective and the sequencing perspective, to incorporate the clustered heat maps, but also others of our tools to help in the analysis of their kinds of data. So I hope that's the kind of interaction we're talking about. We also had an interaction, for example, with NASA, and expect to have more with RICE with relation to applications of what astronomers do well in the big data field and what we do well in the big data field. Much to be learned from that sort of enterprise.
Thank you, John. One more question. Can you comment on privacy protection in bioinformatics, especially in sharing data between institutions? It is important to maintain them even when it slows down sharing. Do you find them unnecessarily restrictive?
Boy, that's a big question. Let me start just with something that's said tongue-in-cheek, especially since we've gone to all the trouble and expense in the institution to install EPIC and the other kinds of administrative and clinical tools and databases we have. If one really wants the security, it seems to me that the way, for example, to handle medical records would be to write them in illegible physician's handwriting, double-sided on onion skin paper, stored in identical manila folders without markings in filing cabinets that were not labeled, in rooms without rooms that were locked and that nobody could find because they had no numbers. As I say, that's tongue-in-cheek in the opposite of the direction we have to take and want to take. I don't know what total security is in the informatics world. Big players still get hacked, but it's been well recognized that that increasingly that the big players who are paying a lot of attention to security may actually be safer. This is conjecture, safer than what we can do locally ourselves, and that in any case, the benefits both to the research enterprise and to patients of sharing is a dominant imperative that we should be pursuing. This is, when we develop software, it has to go through very thorough checks by InfoSec, and we have to be very mindful of any threats. But this is a world of informatics threats, and so that I don't think there's any such thing.
Thank you very much. And with this, John, it was a wonderful talk. We love very much your vision of the science and the life in science. Thanks to all the attendees for being with us for the full period. And in fact, as John was saying at the beginning, please wear masks, simply because this will protect all of us, all of you, and simply because this will allow you to do what you love more, take care of patients, do science instead of being sick. And with this, see you next Friday, 10 a.m. for another great talk by Lima. Have fun, have a nice weekend, and thank you again for enjoying science and being with us.
And thank you, George. Thank you very much.
John, it was a wonderful talk. Thank you for your time again.